Thank you. Thanks for the invitation. So, as with all speaking assignments, I take what I'm offered and then mold a little bit to try to suit what I think would complement what the other speakers are going to touch upon. And so, I thought it would be within my purview to slightly tweak the title here: "The Benefits of Publishing Negative Results," or "The Cost of Not Publishing Negative Results." And put a little bit of a clinical or human impact angle on it, if you will, which is not to say that the others wouldn't, by my estimation or guess, do the same. But being someone who conducts translational research, including clinical trials, and therefore directs a bit of the allocation of resources, by that I mean human resources, in clinical trial investigations, I thought that I would sort of take the tone of why is it that our current publication system potentially does us a disservice in terms of the literature not being populated with the full breadth of experimental outcomes. And by this, I will touch upon both preclinical and clinical instances. Now, of course, it's impossible to talk about the world of non-published data, which is to say all that has never been offered for the currently described publishing landscape to produce for our benefit. But I thought I could at least pull one example from the past few years in my own field that I thought would be illustrative, at least to some degree, of a general point. And then I'll pan back just a little bit more in the later portion of my comments. So I think it's obviously reasonable to say that negative preclinical results can occur from two general categories. One is that the hypothesis is incorrect, which is to say the underlying phenomenon is not there to be observed. And then perhaps the larger and maybe more troublesome one as it pertains to the last question that you asked, which is to say sort of, is this even meaningful data that's being offered to us, has to do with this phenomenon of the experimental conditions perhaps not being quite, quote unquote, right to produce an outcome that was maybe observed by another group. It couldn't be reproduced in your hands, or potentially vice versa. And of course, it's very, very difficult to discern all of this in terms of who's right and who's wrong. Suffice to say that not seeing the universe of outcomes in the published literature has its potential consequences. I think, again, I won't focus so much on the potential underpinning so much as the impact of this data not finding itself into the public domain. So I'm going to focus on an example that has emerged in my specific world, which is quote-unquote molecularly targeted therapy for cancer, and specifically where I exert most of my research efforts in melanoma, where the genetic molecular underpinnings of melanoma largely became elucidated over the past 10 years, leading us to the first successful targeted therapy approach for this population, and that's targeting oncogenic B-raf, an activating mutation that is found in about 50% of all melanomas, and has led to the FDA approval, in fact, of the first relatively selective inhibitor against that target. But the unfortunate immediate aftermath of the initial clinical observation, of which my group was a part, was that just a matter of months later, you could start to see patients lose benefit from that therapy, and resistance would emerge, in fact, relatively more quickly than some of the other sort of oncogene-targeted therapy success stories in a few other cancer types. So a success for sure, because melanoma, particularly in its metastatic state, is a devastating disease and pretty reliably causes lethality within a year's time. So a real problem to try to understand mechanisms of resistance and then ultimately try to overcome resistance. And in our hands, that'll ultimately be a 2 or even 3 drug combination therapy approach to try to essentially intervene against some of the pathways that we have more recently learned can circumvent the initial positive effects. And I'm picking on data produced by one of my very close colleagues and collaborators in the field, because I thought he's close enough to me that for a mixed audience it would be fair for me to do so, because he's someone who I work with very closely still, and have enormous respect for his work. Now, What his group had focused on was culturing B-raf mutant melanoma cells and trying to understand what factors were associated with resistance bred in vitro, which of course is an artificial system that doesn't necessarily reflect the full complexity of human biology, but a reasonable start and a way of trying to get at the mechanisms underpinning resistance to B-raf inhibitor targeted therapy, perhaps in a way that we would have greater difficulty discerning in human patient tumor samples. And so what he found in an initial analysis looking at what were some of the world's very first B-raf inhibitor resistant models bred in vitro was that several receptor tyrosine kinases had their expression upregulated, not new mutations, not the emergence of unique subclones, but rather what appeared to be epigenetic mechanisms that underpinned upregulation of receptor tyrosine kinases. And what seemed to show up reproducibly in some of the cell lines analyzed and across several of the B-raf inhibitors that were used experimentally was the upregulation of the insulin-like growth factor receptor, which is circled as one of the plots in this RTK array in the upper left-hand panel, which was sort of the initial kind of exploratory analysis trying to understand what might be happening within this space. And then in seeking to validate the human relevance of this, patient tumor material was obtained. This is back in the very early days of when B-raf inhibitor therapy was working clinically and then not working some months later, and interrogating patient tumor material to look for baseline receptor expression versus receptor expression at the time of disease progression. And what was seen very robustly in this nice example shown here in the bottom left and in the paper was fairly marked upregulation at the time of progression and really low-level expression at baseline, corroborating what was seen in the cell line shown up above, exposed to two different B-raf inhibitors. This work was then broadened a little bit in terms of incorporation of more cell lines, focusing in here on just one of the B-raf inhibitors for further experiments. And as you can see here, receptor expression goes up fairly obviously in 2 of the 4 cell lines that were analyzed, and further functional work was done to corroborate the significance of this. But back in those days, there were relatively few patient tumor samples available, so they looked at just 5 patient tumors matched pre and post therapy, and only in fact found this one example of upregulation, so 20% with big margins of error around that. In terms of trying to account for how the results may now, in retrospect, have been unique to the experimental conditions used in this laboratory, if you look in the methods section, and I, having worked with this investigator for quite a long time, happen to know a particular fact about how cell lines have been generated in this laboratory for actually decades now, which is that the secret ingredient that allows cell lines to take and be maintained most efficiently is the addition of insulin to the culture media, which is unique compared to most cancer cell line production methods. And so one worries a little bit that that might actually influence the outcome observed here, which is upregulation of the receptor and apparent dependence on it in this in vitro study. Now, the human consequence of this is a paper published in 2010, so rather quickly thereafter, a dedicated phase 2 trial has been launched to combine another MAP kinase pathway intervention, a MEK inhibitor, with an IGFR antibody. And very recently, a biotech company approached me to get my input on the concept of seeking to take their new IGFR signaling inhibitor specifically into this target indication, because figuring that if the biology was pointing strongly in this direction, it was worth them putting a significant amount of their resources behind this being their proof of concept of their agent. As it turns out, multiple groups independently try to reproduce those results, and none of those, and I know of at least 5 of the major melanoma biology laboratories in the world that were unable to corroborate those results, but none of those results made it into the published literature. The only results that speak to the fact that it was not reproducible in other groups' hands were these data where a positive association was found with a different cytokine/growth factor receptor, specifically HGF, activating c-MET on tumor cells. And one of the papers actually was able to demonstrate that it was stromal-derived HGF that was really the product and not a matter of autocrine signaling. But both of these papers actually considered insulin-like growth factor as being a potential mediator of resistance and were unable to show it in a much larger number of cell lines and models than had been interrogated in the previous paper. That wasn't the point of these papers, I should point out, to sort of excuse IGF-related signaling as being a mediator of resistance, but suffice to say that by the by, it was addressed here as a negative result. not the dedicated negative results that didn't make it into the published literature. And part of the opportunity cost here is that we've actually uncovered a number of resistance mechanisms that seem to be at play in this patient population whose tumors resist therapy, in some cases rather quickly. I alluded to HGF, but in fact there are other signal transduction pathways and specific points of intervention for which there are pharmacologic inhibitors available for testing that basically are the other directions the field could be going to, rather than this one. And the issue is fairly obvious, which is that having preclinical data, the positive results only, not the negative results published, leads to the potential consequence of clinical investigations expending real time, capital, and human capital in directions that ultimately aren't potentially as well supported as we might like. And whereas we would all obviously have more enthusiasm for multiple lines of evidence that corroborate the same hypothesis, certainly in the cancer population with patients who have basically no treatment options beyond, let's say, this B-raf inhibitor, where you can imagine that both clinicians and patients themselves are fairly desperate for new approaches, you can understand why the field doesn't sit and wait for corroborating results to be published, but in fact will move on even a single positive or suggestive result.